Thank you very much. So, and also I'm very pleased to see the room filled with many people interested and also with patients. It's very nice to see that patients are also involving here. My interest, the research interest has been since I founded my research group in sialic acids actually, and I started looking for sialic acid as immunomodulators and also sialylated glycans in the diseases like cancer, and then I stepped in the congenital disorders of glycosylation. That's how I was found by Michaela. And one word also for the project is meaningful. That's we are really in the consortium trying to do meaningful aspects, and thanks for all the partners of the consortium. But here what I'm going to show is the contribution of my group for this project in particular. So looking for some efficacy as well as together with the redigers that has already presented before, but also looking for aspects that come from the products which are synthesized just to guarantee that they are not toxicants and they just to reduce their adverse effects, right? So just to among the different products that are being synthesized by Fabrizio, to select those which have the best profile, right? So, okay. So this is the goal, just very general, just to guarantee that we have, that we predict what could be any unwanted effects when effectively they will come to patients when they come, but also to take these in considerations right from the beginning of the project, right? And obviously exclude those which might have adverse effects, right? But this obviously we are just predicting, so this is not effective yet, but then we move to the cells, and then the next steps will be to move to animal models to look for these toxical effects. So this is just an overview of methods that can be used to evaluate toxicity and efficacy of the prodrugs. So in very general what we did, so we have from the knowing the structure from the of the compounds, we can use some algorithms, and this has been performed also with colleagues from the chemistry department in our faculty, and look for algorithms to predict what could be their absorption or toxic effects, and also to look for predict what could be off-target effects, interactions of these prodrugs with other molecules which are within the cells, which we don't know, but according to their characteristics, they might be bounding and may lead to unwanted effects, right? And then obviously we have to test these compounds in cells to look for viability and other features, and also looking for what is a major or one of the readout, which is sialic acid incorporation, right? If they effectively lead to sialic acid incorporation into the cells. And for this, we use lectins or also antibodies, and we do analysis on cells which are defected or not for a GNE. Right? So just an overview. So in the beginning, we received the structure and also the compounds from 5 prodrugs synthesized by Fabrizio. And from this, we did in silico studies to predict what could be this ADMET properties. So ADMET means properties according to the absorption, so the capacity of these compounds to be absorbed. Also, how could they be distributed? I will not enter into details, but given the characteristics of the prodrugs, how effective they will be metabolized, and then excreted, which is also important, and then toxicity, considering potential targets in the liver, in the cardiac tissue, etc. And from this, we had different parameters that you see here, but also some red flags in some of the prodrugs. But for instance, the prodrug 5 has not shown any relevant toxicant effect, so we decided to move with this one, but obviously also testing the other ones, right? But in between, so we also look for structural, so we can have access to critical biological molecules and also simulate if there is any possibility of these prodrugs to interact with these biological molecules. So this is this kind of studies that we do. It's predicted, but it's very important because it avoids us to go for massive studies that can also lead to consuming the prodrugs. Right? And then then we test them in vitro, and we use the hepatic cells just to look if there was any loss of viability. But you see here some prodrugs where we didn't see, with increasing concentrations of the prodrugs, we didn't see any relevant loss of viability, which is good. We also have been comparing this with MANAC, which is, as you know, is already in clinical trial. And then we look for the efficacy in terms of sialic acid. And for this, we use different lectins. So lectins are proteins which recognize sialic acid. And this lectin in particular is a lectin which can recognize different types of sialic acid, which are in the presence of at cell surface, which are alpha-2-3, alpha-2-6, and also alpha-2-8 sialic acid. And you can see here, so we have been using GNE knockout cells. So are cells which do not express GNE. They were given by Videira. And we have the wild type and knockout. And you see that in terms of sialic acid, we have a significant reduction between the wild type and the knockout. And here we have increasing concentrations of the prodrugs. And you see that you have a very nice correspondence. The more we give the prodrugs, the more we have incorporations on sialic acid, right? And actually we see this for the prodrugs that we tested here, these 4 prodrugs. And also we see the same with the MANAC, right? The only thing that is nice to observe is that we can, the fact that we have these characteristics of the prodrugs, we can see the same effect, but with much less concentration, with 10-fold less amount of the prodrugs as compared with the MANAC, which is a good sign, right? Another thing that we want also to address, and this is any potential immune adverse effects. So the relation between sialic acid and immune response has been known, and this is a very simplified picture of how sialic acid could be interfering in terms of immune response, right? So sialic acid, as Videira shows, is decorating different proteins at cell surface. So actually all the proteins which are at cell surface are glycosylated, so they could have sialic acids. And so when we think about very critical immune proteins, including antibodies, so they are also glycosylated and they also have sialic acids, right? And for instance, sialic acids have been described to be very important for the homeostasis, receptor signaling, and also there are other characteristics which are interesting, like pathogens which recognize sialic acids, and also cell-cell interaction. So here represents 2 cells. We have here one expressing sialic acid, the other having receptors, and they are very important immune receptors like Cglex and selectins, which recognize sialic acids. The puzzling thing is that apparently the GNE myopathy patients do not have any immune affectations, but even though we wanted to just rule out if eventually there is an immune trigger that might be on the onset of disease. So we have also been working with other congenital disorders of glycosylation, looking for immune parameters. And it has been interesting because we always find some immune modulation or immune affectation, and that may also be contributing to this exacerbation of disease, right? So, but we don't know, right? Tomorrow we'll know a little bit more with Beatriz, that she was going to present data regarding a very important immune receptor, where we also observed significant difference between the wild type and GNE knockout models. These are just models that we are looking, but interesting, they also responded to the prodrugs which are being synthesized by Fabrizio. Okay, so keep in mind for Beatriz talk tomorrow. So my role also in the project is to take a guarantee that we also take the prodrugs and we do all the steps to have a product at the end of the project, which is the target. We call it the target product profile, right? And for this, we need to have also data from how the products will be distributed. And for this, we have been doing the pharmacokinetics study. So for this, it consists of having an animal model, which is a healthy animal model, but has been treated with the prodrug that we have been selected. And now we collected several organs, but also urine and serum, just to make sure to know how the prodrugs will survive, let's say, when administered to the patient. So this is very important because we want to know if it takes hours, days, or minutes, because this will be important when we consider the dosing and also the frequency of the dosing and the patients. So with this, we also, as I was mentioning, we are looking for what could be the final profile of the prodrugs. So obviously we are in the just very early steps, and it has been interesting because we have been some advices from EATRIS, so the European infrastructures, also that has also been helping in mentoring these projects that is funded by the European Joint Program. And so we believe that this is just already reasoning what could be the final profiles, right? So we want a drug that could be obviously having effect on the muscle strength, improve the muscle strength, but also having changes in biomarkers. And that's why in the project we have a team dedicated to biomarkers. And the route, as already was mentioned by Luca, the route that we perceive will be the oral route, and ideally with less than one daily intake. So this is what we think it could be the profile of our product. But that's it. So this is very key that we think about all these considerations, but also that we put the patients and also their opinions in the first place. And that's why that's been very instrumental to have Michaela leading also this project. And this is very important also that we think how it could be acceptable and usable for the patients. And there are also, it's very important that there are other students that have been somehow involved on GN myopathy. So I think a nice outcome is really to have more awareness of the disease itself, right? And with this, grazie mille, muito obrigado. Thank you.